Our next speaker is the Deputy Director for Science and Head of the Materials Department at InnoRenew in Slovenia, as well as an Associate Professor at the University of Primorska and the Management Board member of the new European Bauhaus Academy Pioneer Hub. In 2022, she was awarded a European Research Council Consolidator Grant for Archiskin, a project pushing the boundaries of engineered living materials. Her research on probiotic architecture continues through the Remedy project, recently funded by the European Innovation Council under the Pathfinder call. Ladies and gentlemen, please welcome Dr. Anna Sandak.
Okay, thank you so much, and thank you, Bence, for the invitation. I really feel honored to present you what we do. And today in morning we hear how hard it is to be astronaut and to be selected. But I believe that every one of us has also a chance to contribute a bit in the space science by our development regarding the research we are doing. So I will present you a bit of flash of my project Archiskin, but I will also present you what we are planning to do together with Bence with the Mission Possible. And then my core business is the building sector. I work with the building materials, and my focus is on renewable building materials. So I will just challenge you a bit with the material science. Then we will just think if we need alternative for what is going on now. And then I will introduce my topic, engineered living materials. Then also present you a bit of the Archiskin project, and then I will finalize with the Mission Possible outcome. So when we think about the materials, I think I will not say something wrong, but I think materials shape the civilization, because if you are just thinking about the timeline, there is so many of materials developed in the time, but there is still questions mark. So is the space for us to develop something new? But when we think about the building sector, still the most common building materials is the concrete, with 10 billion tons of concrete produced every year. So that's really huge amount. And also we produce a lot of plastics, so this you also know. And some of these plastics are ending in the sea, and I think you all hear about the microplastic, nanoplastic, and at the end those materials are ending up in our body, unfortunately. And then in 2020, when we are just thinking about the materials generated by the humans, like the concrete, plastic, asphalt, metals, and so on, everything what human produce exceeded everything what is living in the biomass. So that's really that was the point when everything living was less intense than the building the materials. And then there is also this boundary conditions for our planet. There is 9 of them, and we already crossed 6. So that is really point that we are shaking the equilibrium of our planet. So this is not and we already see the consequences. And the consequences are here. So we have the extensive drought in Africa, and that was the photo from the Balkans in this winter. It was an extensive amount, exceeding amount of snow. In Bologna, they had flow. And then I think you hear about what was going on in Hollywood, the fires, which is very hard to shut down. So we are shaking our planets, and then we are putting this to really extreme. So this word will come up also later on. And then, but we are more than 8 billion people on the planet, so we still need materials to build the houses and to accommodate the population, which is continuously growing and growing. But maybe, just thinking about in the general perspective, maybe we need alternative and then new perspective on sustainability. And then as biologists from the background and then material scientist, I always look at the master, so what we can do. And I really like this nature-inspired approach, because nature already developed materials for 3.6 billion of years. And as humans, as humans on the planet, the Homo sapiens is 300,000. So I think we have big master to learn from. And we're just thinking about the materials generated by the humans. So we are making the assembly, complex materials with a lot of volumes. Very often we are using extreme conditions like the high temperature, high pressure. And the production, even thinking about the circularity, we still have a lot of linear production. At the end, we have disposal. So we are creating waste. And in the contrary, when we think about the nature, so nature is has different approach for building materials because it's by the grow. And the simplicity There is more about shape than the volume, and the processes are done in the ambient conditions, and the production is really circular because the waste from one organism is the nutrient for another. So we are ending up with the decay. So this is something to consider. And when we think about the biological system, there are two fascinating properties because it's multifunctional, and I often giving the example of, for example, of our skin. So we have skin is acting as our barrier, so it's protection. But skin is also a regulator, and at the same time, it's also sensor because we are sensing. And very often, skin is also signaling because we can see, okay, we are stressed, we are white or red, and so on. So it's multifunctional. And also, biological systems are highly optimized. So there is nothing like which serve without the purpose. Everything is needed. So that's really nice properties. And then, of course, bio-inspired innovation, I'm working in materials field, so of course we can be inspired to generate new materials, but this is not the end of the story. Also, we can be inspired to generate new shapes of materials, or the way organisms are perceiving can inspire new generation of the sensors. The way organisms are moving and interacting can inspire mechanics or kinetics. Then also the way of organisms are communicating can also inspire new way of the communication for us. And finally, they can inspire our processes. But can we go beyond of that than bio-inspiration? And what is our vision for materials development? As I was mentioning, I'm a biologist and material scientist, so we are trying to merge these two disciplines which are not very close by default, and to replace sustainability, which I was mentioning before, with regenerative design. So it's something which is positive for us. And then design with nature is absolutely wonderful because we can be inspired by nature, we can be informed by nature, but we can be also empowered by nature, and that's really, really powerful. And then when we think about materials in nature, they have these self-properties like self-regeneration, self-healing, and so on. So just if you think, what if materials with such characteristics can be made? And what kind of new application we can imagine if we have these self-materials generated? And then comes the topic which I'm working with, these are engineered living materials. So those are materials which have embedding the living cells in their shape. And then those cells can be different. We are working with fungi, with algae, with bacteria, but can be also mammalian cells. But in simple words, we are adding new dimension which is not existing for materials. We are adding life, and that's really cool property. So what are the key features of engineered living materials? They have the regeneration and self-healing properties, which is really hard to achieve with conventional materials. They have the growth and adaptability. They are, of course, environmentally responsive because they are interacting, they are resilient. And finally, they are also circular and sustainable, so that's really perfect combination. And I was just wondering what properties of the materials can be adjusted for the building sector. And here I was listing some of them, and those are the properties which are highly interested for the building sector, and those are possible with the living cells. But if you translate all of this to the house, so we can have the house which can self-repair themselves, can be adapted to environment, can detect some errors and can sequester the carbon and produce the oxygen. So that's really fantastic. So we will maybe in the future, we don't have to maintain our houses because they will be growing by themselves, you know? And then just going back to the topic I'm working, so I'm working with the coatings, which is like, coating is something you are applying on the building facades to provide the decoration, but also to protect the materials. So those are the numbers, so it's like the huge sector, and of course, having in mind that new buildings will be built in the future, this is continuously growing. But of course, our houses are exposed to different environments, and this is very often what we are facing. So we just think, okay, it's just the climate, but very often, again, this is very, very extreme. And then, so everything what is outside, what is built outside, is exposed to the weathering. And this is natural process we cannot somehow shut down. And this is the combination of the biotic and abiotic factor. And at the end, very often, the coatings are looking like that. So this is not what the architects or clients wants to have, because it doesn't look nice and doesn't protect anymore the materials, yes? So what is our approach, what we do as a human? Of course, let's make like, let's put some chemicals, biocides, and like, okay, we are doing something which is maybe a bit long-lasting, but at the end is very toxic for the environment, and at the end of life, we have trouble to recycle such things. So what we try to do, we are trying to use the nature. So the concept I'm trying to push for is the probiotic architecture. So I will explain what is this. So we are trying to integrate the living system in the materials, and then to have the healthy ecosystem with the symbiosis with the different microorganisms, and then to have this microbial interaction. So this is our and then you can see here the petri dishes of our fungi, but we are working with the microorganisms which account 2/3 of life in the earth. So this is like, something which is not visible, but doesn't mean that doesn't exist. So, and then when we think about our body even, like if you count the cells, we are more microorganisms than the humans. So that's something really good to consider. Something what is not visible doesn't mean that it's not existing. And then coming to the topic. So this is the project when I was got the grant in 2022. And Archiskin, when we try to have this concept of bio-inspiration, living cells, and then this nature-inspired mechanism to develop coating system, which is serving for different architectonical surface, can be concrete, can be steel, can be plastic, can be wood, and providing such properties. And to do so, we are optimizing engineering biofilm. What is biofilm? This is the same photo of your dental plaque. So, okay, it doesn't mean that we are making dental plaque on the building, but just to explain what is the biofilm, this is consortium of different microorganisms which are all together. And of course, as you know, when we are all together, we are stronger than one single cells. So they are like living all together, they are providing the nutrients for themselves, and then they are very, very strong. So for doing this, we are using fungi, and this was like very interesting. Today we had a lecture about the algae and about different microorganisms. We are using fungi, and fungi are absolutely fantastic. So there are like 120 species which are identified, but so many still not discovered yet. And then when you just look at them, they are like really beautiful. We have fungi which are bioluminescent. We have fungi in the gorgonzola cheese. By the way, in the beer and wine is also because of fungi, and our bread is because of the fungi. We have also psychedelic fungi, but maybe this is another project. But nevertheless, there is so big portfolio of species which is like really unexplored. But by the way, the biggest organism in Earth is also fungus, which is growing and like has 10 kilometers square, like the Haifa in Oregon mountain. So what we were thinking, okay, let's see what the Mother Nature is giving to us, because we didn't want to introduce something which is like not present. And then we started from the investigating local microbiome in Isola, and then also assessing bioreceptivity of different materials. So bioreceptivity is ability of the materials to be colonized, but without degradation. So that was the point we wanted to achieve. So we were sampling with the students different fungi. And then we were running two different research lines. One was characterization of materials, because we wanted to assure they are not degraded. And the second was this microbiology line, because we wanted to understand which organisms we have in our portfolio. So that is the example of characterization with different microscopy, perspectral imaging, and so on. And also this molecular identification, just to understand with the DNA analysis which exactly species we are dealing with. And this is what we found, and then you can see those beauties also on the table here. We have this is what we found in our microbiome around the place, and we were thinking, okay, let's select something which is very common, because we would like to have coatings which will be not just working in our place, but is in different conditions and so on. So our superhero is Aerobasidium pullulans. So this is our selected guy. We selected that one, and we had some reasons why to select this. Is very ubiquitous fungus. So it means it's growing in different, you can find it in the desert, in the glacier, in different environments. So it's everywhere. It's polyextremotolerant. It means can survive very different conditions. Very low temperature, very high temperature. High humidity, low humidity. So it's very tolerant. Has a high phenotypic plasticity. What does it mean? It can switch from one growth of mode to another. And when the conditions are not favorable, it will turn to chlamydospores and will just wait. It will be hibernated. It will not grow, but will be still alive. So this is also capacity which is very, very important for our application. Then it has also broad enzymatic profile. What does it mean? We can feed with different nutrients. At the same time, it's oligotrophic, so it means then doesn't need much of food. But we can provide different nutrients, it will be still growing. So I was observing you a bit like how you were eating the lunch today, and we were like trying this, trying that, and then this is really nice to enjoy the food. But in microbiology, this is very, very simple because the organism will not waste energy for something which is very hard to digest. It will go for the simple source. So we were having this in mind. It's really nice to understand this because we can feed the fungi with what we want to feed him. Has also brought pigmentation. You can see here the fungi are also developing pigments by themselves, and this could be very, very colorful. For the architecture, this is very important because we would like to have facades which are colorful. And finally, it's not toxic. We were testing the fungus with the epithelial cells and fibroblasts, and there is no toxicity for the human. Of course, this is very important. We would like to have something which is not harmful for ourselves. And this is our Aerobasidium pullulans with the microscopy. So these brownish cells are these chlamydospores which survive the extreme conditions. So perhaps in the space they will be in this mode of the growth. So what we were trying to do, okay, we have our candidate. We would like to be able to apply it as for the coating. We would like to understand how it grows because we would like to manipulate somehow. We would like then the fungus will grow in the way we would like them to grow. So what we were thinking, okay, let's test the different nutrients. So we were growing the same strains on different nutrients, and we saw then the cells, the types, the morphology was completely different. So this is the way to manipulate the growth. Then we were adding some additives, and we also saw then for just adding a bit of hitosan, the branching were very deep and going very penetrating the material. So this is the way also to manipulate, for example, to speed up the growth. Then also we were testing different strains. So when we decided for Aerobasidium pullulans, we said, let's see, because different strains have different properties. And so we tested at the beginning over 60 different, and then we narrowed down to 10, and now we are working with 3 for the most promising one. And then here you can see the time-lapse microscopy, so observation how the fungi are growing. And you can see then from on the left you have much more hyphae, and then on the right is much more the yeast cells. So and this is the same strain, but just growing with 2 different foods. So just having this in mind, we can really manipulate the growth. Then we were also thinking, okay, for us, I was telling you about the biofilm. This is a very important part of our research. We were testing different strains, and then we selected the one which is producing the biofilm by itself, with the quantity which is suitable for our application. So this is also varying. That at the end, we were thinking, okay, if we will just observe with our eyes, this is very biased and subjective, so let's make some modeling. So we were also doing some mathematical modeling to model the growth of the fungi, just to compare which is really faster and which is the most branching and so on. But going to the topic now for the conference, it was mentioned already in the morning, then when we just looking at how much does it cost the launching something in space, of course it costs a lot. The prices are going down, which is good for us because making this research affordable, but still it costs. So this is the question, take or make, because we cannot also take everything to the space. So maybe there is also option to produce something there. And then, so what if materials in space can grow and also repair themselves? And then just as living organisms. So maybe this is also the way for materials to require less maintenance and less input from the humans. Okay, so how can we boost engineering living materials development? So what we do in the laboratory is design, build, test approach. So this is the standard approach we are doing when we are developing new materials. But having in mind mission possible, I was thinking, okay, what doesn't kill us, make us stronger. Okay, let's launch our fungi in space. Okay, we will not have the interaction with bears, so it will not kill us. But this is maybe the way to boost the development, and whatever will come back maybe will be much more resilient than it was before. Yeah, so this is the idea. Okay, let's challenge our fungi to the environment, which is not the standard one. And then that's the mission possible. I think this was shown so many times today, so I will not repeat. Okay, and we are really much looking forward to see what will come back. And then, of course, it's not coming with Matt Damon to the Mars, but still we are expecting to have the microgravity, high radiation, limited nutrients, and oxidative stresses. So this is, I really hope this will somehow affect the growth, and then we will see what is before and after. And of course, extreme conditions. And I was just reading last week the very nice paper in Nature, and then when was stated, and then what was about microbes in space, and the design of experiments can make the difference between success and failure. So I really hope what we are sending, it will be successful, because if not, we will also learn the lesson. But nevertheless, this is our experiment. So this is Aerobasidium pullulans, the strain, which was like producing quite amount of the biofilm, so it's also on this table. And then it was packed in this payloads, and is now in the Mayasat waiting for the travel. And then so, of course, we have the control samples stored in the fridge, and original strain stored in -80. So this is like the ultra low freezer, just to not change anything, and the 5 payloads which will be integrated. And what we are planning to do, so this is the microscopy, environmental SEM microscopy and fluorescence with some trackers, just to see what the morphology is changing. Then we will do whole genome sequencing to detect any mutation, also RNA sequencing to analyze the gene expression. Then we will also make some experiments regarding the stresses under UV radiation and the different temperatures and nutrients, just to see if the growth is somehow changed. And then also we will co-culture with pathogenic microorganisms, just to see if it's much more resilient than the strain we sent. And finally, also we will track the metabolic changes with the GC and LC mass spectrometry. So what we are expecting to have, so we hope to have some differences, of course, in the biofilm formation and morphology, with the phenotypic plasticity, maybe some mutation which are linked with the survival. Maybe there will be like some extreme tolerance shift, and then maybe also epigenetic regulation. But at the end, we would like to have material with the strains which are coming and will be more resilient. And then finally, to use this for this application of engineering living materials. So what is the role of engineering living materials in the resilient architecture? So we are integrating synthetic biology and then these living interfaces to produce materials which are much more environmentally friendly, and also having this capacity for the self-healing and lower maintenance. And then, of course, having in mind the resource efficiency and then not generating waste. And finally, to have this for our benefits. So this experiment, I hope, is not only used for our curiosity, but I think really these experiments can, of course, boost the materials development for the construction, but also for the aerospace industry, for the defense, for the health applications, and of course, in space. So when we just think like which materials we can expect for the Earth application, I hope to have something which has better durability. Maybe materials will be lighter because they can be manufactured in different way. And then also this manufacturing mode is different. And finally, we hope to have them more sustainable. So this is my team. I would like to acknowledge the ERC for sponsoring the grant and also making our dreams come true. And thank you again, Bence, for the vision and making these things possible also for the researchers. It was today very nice overview for the grants, how much Europe is putting money for the space travel. I quite follow the European Commission calls, and just to let you know, then in spring there will be new calls launched from the Cluster 4, Digital Industry and Space. So and then already the draft program is circulated, so we can have a look what were the topics which the Commission is prioritizing. So the calls will be closed in September. So also for the researchers or for the developers, for the companies, there is to benefit from the funds. So that's all from my side. I will be happy to answer the questions.
Yeah, Anna, thank you very much. The wonderful world of fungi. No vegetable, no animal, but fungi. Do you have any questions, ladies and gentlemen, from the floor? Anna is more than happy to answer any question. Oh yes, of course, our colleague. Sorry. That's okay.
Thank you again. So yeast-to hybrid system, have you considered using any protein-protein interactions and including into your assays yeast-to hybrid systems? Because perhaps biochemists, molecular biologists would be loving to see some of the key proteins which could be benefiting from this trip.
This is just the beginning of the story, I believe. Okay. We are now, for the moment, working with the fungi only, but we already have we started 2 weeks ago a new project when we are making the consortium of microorganisms together with the algae and bacteria, so we're making the things more complex and complicated. So depending what will come back from the space, if we'll be able to see any difference, of course we will go deeper and deeper, and we will try to see very profoundly what was changing.
So first, let's see if they survive.
Yes.
Okay.
Yeah. Let's hope they survive, and if yes, then we will do everything what is possible.
Thank you. Another question from the floor. Thank you.
Thank you. So yeast is also on board.
Yes. This is yeast like fungi. Yes. Yes.
But our yeast as well, because we also send yeast. I was not talking before, but my colleagues also gave yeast. So, we can think about to maybe do the same analysis on all these samples who will come back.
Okay, of course. Thank you.
Thank you. Any more questions? No. Lovely. Anna, thank you very much for your presentation. Thank you.